Right. In the last session, we discussed 2 questions and we were discussing the third question. The centriole in the head is the proximal centriole. Centriole in the neck. Centriole in the neck is also the proximal centriole. The proximal centriole extends from the head into the neck region. The tail is formed by the distal centriole. Tail is formed by the distal centriole. Acrosome is formed by, or it originates from the Golgi body. Acrosome is a modified lysosome. Now pay attention to these questions. Endoplasmic reticulum. Now this is from class 11th. Endoplasmic reticulum forms Golgi body. Endoplasmic reticulum by means of its vesicles. By means of its vesicles form Golgi bodies. Right. Golgi body produces vacuoles. Vacuoles having the hydrolytic enzymes. Which enzymes? The hydrolytic enzymes. Right. Now these hydrolytic enzymes containing vacuoles are basically the lysosomes. It also produces vacuoles having secretions, the substances which would be secreted to the outside. Right. Now this lysosome in this case becomes the acrosome. It becomes the acrosome. Okay. Hence we say acrosome is a modified lysosome. It originates from the Golgi body. Right. Complete spermatozoa. Complete spermatozoa enters the ovum. The ovum. Complete spermatozoa enters into the ovum. However, only its nucleus enters the cytoplasm. Of the female gamete. These are the questions which are asked in various examinations from the part which we have read till now. तक हमने फर्स्ट स्टेप पढ़ा अप्रोक्सीमेशन, ठीक है, विच इज प्रीसीडेड बाय कैपेसिटेशन. कैपेसिटेशन का मतलब समझने की कोशिश की हमने, कैपेसिटेशन क्या होता है, ठीक है. एंड विद दिस वी स्टडीड द स्ट्रक्चर ऑफ द स्पर्मेटोजोआ, राइट. नाउ so basically in this manner the first step became capacitation. The second step is approximation. Now the third step is agglutination. The third step is agglutination. This is fixation of spermatozoa to the surface of the ovum. Right. Now the ovum. Ovum is something like this. Let this be the ovum. Right. Inside this is the secondary oocyte. Inside this there is the secondary oocyte. Right. Just outside this secondary oocyte is a layer of chemicals. If you remember, we studied the structure of Graafian follicle, mature Graafian follicle, right? So there is this layer made up of the substances. Then outside this, just outside this, is a layer of radially arranged cells. The cells are arranged radially. इस तरह से लंबे लंबे यूं लाइंस में अरेंज होते हैं. से एक सेल ऐसे है, फिर दूसरा सेल ऐसे है, इस तरह से. This layer is known as the corona radiata. The corona radiata. The corona radiata. The layer inside this, this blue, the layer of chemicals is known as the zona pellucida. इसे हम क्या बोलते हैं? This is the zona pellucida. Right. The membrane of the secondary oocyte is known as the vitelline membrane. The vitelline membrane. This structure is the secondary oocyte. This is the secondary oocyte which is formed from the primary oocyte. प्राइमरी oocyte से सेकेंडरी oocyte बना होता है, ठीक है. तो सेकेंडरी oocyte के साथ एक छोटी पोलर बॉडी भी बनती है, अगर आपको याद हो, which is the first polar body. Where is this polar body? The polar body is situated here. यहाँ पे. This is the first polar body. Right? Okay. Now this is the secondary oocyte and this is the first polar body. Now to this is attached few cells of that stalk that we talked about in the follicle. यहाँ पे अभी भी कुछ cells क्यूमुलस oophorus के attached रह जाते हैं. This was the stalk to which this structure was hanging inside the Graafian follicle. If I just make you revise this, theca externa, then there was theca interna, theca externa के बाद theca interna, ठीक है, और theca interna के बाद membrana granulosa, the third layer, and membrana granulosa was surrounding a cavity. And from one part of this membrana granulosa, there was a stalk which was hanging down. इस तरह से स्टॉक थी, ठीक है, और इस स्टॉक से कोरोना रेडिएटा, जोना पेलिसिडा इन स्ट्रक्चर से घिरा हुआ सेकेंडरी oocyte. और इसके साथ में यहाँ पे ये छोटी पोलर बॉडी. Right. So basically, this structure comes out of this. This structure is this one. This is the complete follicle. This is the Graafian follicle. This is the Graafian follicle. Now from this Graafian follicle, this structure is actually released out. It breaks from here and comes out. It pops out. बाहर को निकल जाता है. ठीक है? Which is the ovum. So these are the cells of. This is the cumulus oophorus. Cumulus oophorus. The cumulus oophorus, corona radiata, and zona pellucida are the coverings of the ovum. Are the coverings of the ovum. Right? These are produced by ovary. These are produced by the ovary. Whereas this fourth layer, the fourth covering, is produced by the egg itself. Vitelline membrane is the plasma membrane. Vitelline membrane is the plasma membrane, cell membrane of the secondary oocyte. Right? This is produced by the egg itself. The egg itself. So the membrane which is produced by the egg itself is known as the primary egg membrane. The primary egg membrane. And the membrane which is produced by the ovary, which is secondary in origin, this is secondary egg membrane. आपको ये क्वेश्चंस भी पूछे जाते हैं, प्राइमरी एग मेम्ब्रेन और सेकेंडरी एग मेम्ब्रेन क्या होती है, इनके एग्जांपल्स बताइए. प्राइमरी एग मेम्ब्रेन जो एग खुद बनाता है, जो एग ने खुद ने बनाए हैं, एग इटसेल्फ. सेकेंडरी एग मेम्ब्रेन जो ओवरी बनाती है. चार तरह की मेम्ब्रेन्स हो सकती हैं. फर्स्ट मेम्ब्रेन जो एग खुद बनाता है, सेकेंडरी मेम्ब्रेन विच इज प्रोड्यूस्ड बाय द ओवरी. इसके अलावा कुछ एनिमल्स में इसके बाहर भी एक मेम्ब्रेन होती है विच इज प्रोड्यूस्ड बाय द रिप्रोडक्टिव ट्रैक्ट. लाइक फॉर एग्जांपल द एग वाइट. हेंस एग में, मुर्गी के अंडे में, हेंस एग में there is this egg white. It is produced by the oviduct. It is produced by the oviduct. Now that egg white would be the tertiary egg membrane. So human egg is covered by two egg membranes, the primary egg membrane and the secondary egg membrane. Right. Now we have to study this. Why? Because we have to study acrosomal reaction. The sperm has to cross all these barriers. The sperm has to cross all these barriers and then it would be finally in contact with the vitelline membrane and then it would release its nucleus. Right? So spermatozoa has to cross all these membranes, remember. So the process of agglutination is fixation of sperm initially onto the surface. Right? The spermatozoa gets fixed onto the surface of the ovum. This is known as, this fixation is known as agglutination. इस फिक्सेशन को हम क्या बोलते हैं? एग्लूटिनेशन. ओके. The reason for agglutination. The reason for agglutination. Fertilization anti-fertilization reaction. Fertilization anti-fertilization reaction. This is responsible for agglutination. Fertilization and anti-fertilization these both are glycoproteins. They are basically glycoproteins. Chemically, glycoproteins. Carbohydrate plus protein, glycoproteins. Right? Okay. The fertilization is present on the egg. Fertilization is present on the egg surface. Fertilization is related with the egg. Anti-fertilization is related with the surface of the sperm. Fertilization on the surface of egg is highly specific for anti-fertilization on sperm or spermatozoa of a species. Hence, the gametes of two different species cannot fuse with each other. तो अलग अलग स्पीशीज के गेमीट्स आपस में फ्यूज नहीं कर सकते. ठीक है, इसको मॉडिफाई किया गया है. Gametes of same or closely related species can fuse. अब एक क्लोजली रिलेटेड का एग्जांपल क्या है जी? हॉर्स एंड डंकी. The equus, which is the horse, and Equus domesticus. The progeny is known as mule. Panthera leo. That is a lion. And Panthera tigris. That is the tiger. Right? There are two offsprings, liger and tigon. So sometimes the gametes of the two closely related species may also fuse, but that specificity lies upon the fertilization anti-fertilization reaction. इस चीज पे डिपेंड करती है. फर्टिलाइजन एंटी फर्टिलाइजन अगर मैच कर जाएंगे तो गेमीट्स फ्यूज कर सकते हैं, अदरवाइज गेमीट्स नहीं फ्यूज करेंगे. Fertilization is present on the surface of the egg and anti-fertilization is present on the surface of the spermatozoa. This is the reason for agglutination. This is why the spermatozoa gets fixed onto the surface of the egg.